Hi everyone, welcome back to the channel. This is Dr. Asa here. Today's topic is review unit number 2 in BSC1010 lab. So please remember to keep up with the resources available to you already, your syllabus, lecture PowerPoints, anything that any notes that you took. Read your reviewing notes in the class and the experiments also you did during the last 3 labs. So remember guys, we have three major labs we did in this unit 2. So they are membranes, lab 4, lab 5, enzymes, and lab 6, photosynthesis. So let's get started. Okay, so the test is gonna cover just these 3 chapters. So the membranes, lab 4, we talked about especially the membrane transport, and all our experiments were related to membrane transport. So remember, membrane is a selective barrier which actually has transporter proteins. They help with transporting stuff in and out of membrane. And also we use simple diffusion, moving stuff from high concentration to low concentration using concentration gradient. So always remember diffusion and osmosis, right? Semi-solids, Lodia, our plant, potato cells, red blood cells, some of the materials for membrane lab. So then if you remember, so we had 3 different situations possibly could happen. So hypertonic, which is if there is a higher outside solid concentration than inside cell, that's an hypertonic situation, which is basically salt is gonna move from high to inside low, and then water is gonna move out from the cell. That's a typical hypertonic situation. This will happen until it reach what we call isotonic, which is basically the both sides reach same level or equilibrium. So remember we did the experiment with potato cells, and we use 6 large test tubes with the potato cubes in them, and then we had this differential concentration of sodium chloride in them, and we try to find out which test tube is going to give us the isotonic, or which point exactly is isotonic point. So in order to have, also another thing here is, in order to have these nice curve there that actually to find out exact point of isotonic point, so we use something we call serial dilution. So serial dilution is you have a stock solution, and then you're sampling by diluting it in a series of dilutions, basically, and then to find to get this series of test tubes with different concentrations in it, all the way to the 0, so that you can actually have a nice curve to calculate. Right, so it's not just specific to here. We use this same serial dilution in next lab, which is Enzymes Lab. So serial dilution also applied here in Enzymes Lab in order to find out specific concentration of the enzyme or optimal concentration of enzyme. So we use enzyme malase here, salivary amylase, to find optimal concentration, optimal pH, optimal temperature. Always remember, enzyme and substrate is like a key lock model. So every enzyme has a substrate, vice versa, and the enzyme get in reaction with the substrate and then complete the reaction and then moves out and to the next reaction. So, but enzymes are very specific about what temperature they work, and what pH they work, and what concentration they work. So we did a series of experiments to test to find out best color change for this enzyme in terms of what temperature, what pH, what concentration. So remember, we are only working on one enzyme here. So that means every enzyme is unique in terms of their temperature, pH, and the concentration. So, and always remember, enzymes are proteins. So anything applies to proteins, apply the enzymes. So including denaturation, if they actually in extreme conditions of high temperatures or pH, they can denature as proteins and they go back to their primary structure. Okay, so that was lab number 5, which is enzymes. Lab number 6 is photosynthesis. Your book calls it energy transformation. Basically, photosynthesis is the most important reaction on biosphere. So it's carried out by plants and some algae, green algae, and a couple of bacteria, which they all have one thing in common. They have chloroplast, chlorophyll, and they have chlorophyll, and then they can carry out this reaction. So remember our reaction, we must have CO2, we must have light, we must have chlorophyll, water, and then our products are oxygen and water. Right? So this takes place in plant sense. So we use a number of plants in this lab, including geranium, spinach, begonia. Not begonia, coleus. And also as well as our plant, aquarium plant, Elodea. So we have a series of 5 major experiments we did in this lab, in photosynthesis lab. And always remember, we can test light using the spectrophotometer. We can test CO2 using phenol red. We can test chlorophyll and also other pigments using chromatography. And also we can do a light and dark experiment using a plant like geranium. So you remember the plant names, remember each of the experiment, remember how we tested them. Eventually, we actually indirectly measuring the starch accumulation and with the Benedict solution and color change. So always remember the correct color changes in all labs, regardless what actually color change you might have seen in your own experiment. So always remember that. Always do some flashcards, prepare some flashcards, redo your notes, review these 3 labs, and just to recap again, this is module number 2, review of the labs 4, 5, and 6. And again, please feel free to contact me with any questions, or come by office hours if you need extra help with the material. See you next time. Best of luck with the material and studying. Happy studying.